Hello everyone. This tutorial is for exercise 1, survey of invertebrate evolution, part 1. So before we start talking about invertebrate animals, let's talk about what an animal is. They are eukaryotic, meaning that they have cells with complex organelles and linear chromosomes. They are multicellular. They are heterotrophic. That means they can't make their own energy from the sun. They have to get it from elsewhere, generally from eating other animals or plants. And they have no cell walls. Many of them have some sort of sensory cells, meaning that they can sense their environment, and muscle cells, meaning that they can move. Most are diploid, meaning they have 2 copies of every chromosome, and they reproduce sexually, producing haploid gametes. Some will reproduce asexually, some are hermaphroditic, and some combine 2 or 3 different reproductive strategies. So we can further characterize animals by type of body plan, right? So we can determine how they relate to each other phylogenetically based on what their body plan is. We got 2 basic organization of tissues. We got parazoans that have no true tissues and eumetazoans that have true tissues. We'll also talk about classifying them by germ layers, their type of symmetry, their embryonic development, their body cavities, and their segmentation. So all of our animals we're going to talk about today presumably developed from a chanoflagellate ancestor. All of our eumetazoans, so the ones that we talked about that have actual tissues, all the ones that, so excepting peripera, which are ones that don't have true tissues, have some sort of symmetry. So our hydra here has something called radial symmetry, meaning that you could cut it in half along these lines, each of these lines, and get an equal half. All right. A lot of other organisms we're going to talk about have bilateral symmetry. So that is our first split here, right? So we have all of these animals that show some sort of bilateral symmetry, and then we have our radially symmetric animals here, our ctenophores and our cnidarians. So our eumetazoan animals also have germ layers. Basically what that means is as they develop from a single-celled egg and the cells divide and divide and divide, you form basically this hollow ball. It's a single layer of cells. That then starts to indent, and those indents the layers that indent will form layers of the organism. So we end up here with 2 layers, right? We've got gastrulation at first opening, and then we end up with 2 layers, the endoderm, which is the innermost layer, and the ectoderm, that is the outermost layer. So what you end up happening with these guys is that our two types of symmetry also line up with our 2 different types of layering. So we've got our ctenophores and cnidarians that have just an ectoderm and an endoderm, all right, with sort of gelatinous material in between. And then the rest of our critters all have the endoderm and ectoderm, but they have another layer called the mesoderm, all right, which is going to develop into other things as the organism matures. Okay, so those organisms that have a 3-layered system can be further divided based on their embryonic development. So one of the big things is the fate of the blastopore, which is that first opening, that first little indent that happens. In our protostomes, the first opening that shows up is going to eventually form the mouth of the animal, and in deuterostomes, the first opening that forms is going to become the anus of the animal. So our deuterostomes, the ones where the anus forms first, that is us and chordates, and echinoderms, which are sea stars and sea urchins. Protostomes are basically all the other ones. So arthropods, roundworms, annelids, mollusks, flatworms, rotifers, and lophophorans. Our protostomes can be further divided by the type of larva that they have. All right, so if their larvae at some point during their development have two bands of cilia, they are called trochophores because they have a trochophore larva is what that is called. So all of these guys are going to have that trochophore larva. And then lophophores, where the larvae have a fan of ciliated tentacles, and the only one we've got there are lophophorans. So the next way that invertebrates are divided up into groups is segmentation. So some of them do not have segmentation. Some of them have segmentation where each segment is relatively identical to the others. All right, so some of our roundworms are like that. They would be the same inside and outside, meaning that the segments look pretty much the same on the outside, and they would have the same organs in them. In fact, a lot of the organs would repeat as you went along the animal. In other animals, the segmentation differs internally and externally. Right, so we get our classic sort of shrimp diagram here. You can see that there are different appendages on each of the segments, and the shape and size of the segments is different. The internal organization would be different as well. Circulatory systems, right? So these aren't super useful for classifying the animals because some phyla have more than one approach to this. But one of the simplest ones is the gastrovascular cavity. Basically, you've got a cavity in the middle of the organism. Water can move in and out of it to help bring oxygen. This is also going to function as the digestive system of the organism as well. There is only one opening where circulation is going to happen both into and out of the animal. More complex is the open circulatory system. So we've got here insects and we've got mollusks that are showing an open circulatory system. Basically what that means is, like you can see here dorsally, there is a heart that pumps blood. That blood then just flows out into the body cavity, sort of oozes its way through the body cavity before being picked up by the heart again and recirculated. So that means that when this blood is in the body cavity, it doesn't really have any pressure of its own. It only gets sort of pushed along by the locomotive muscles of the organism. Skeletal systems. So some of them have none, right? We've got peripheria, which are the sponges that don't have a true skeleton. They do have spongin, which is sort of a support matrix that holds them up, but it's a protein support matrix that holds them up. They also have spicules. Here's a few different examples of different kinds of spicules. These are made out of calcium carbonate, which is very similar to sort of mollusk shell, or some of them are made out of silica, which means they're basically biological glass. Right? So they're very stiff, and they help provide some support and also protection against being eaten by other animals. You've got a hydrostatic system. All right, so how does a hydrostatic system work? Basically, you can think of it as Olaf here in his Macy's Day balloon. He doesn't have any sort of internal skeleton. There's no external skeleton. He's made rigid by the fluid, in his case, helium, that is pumped into him. And in the case of our rotifers and our cnidarians here, it is actual water that is pumped into them. And the pressure of that fluid inside them is what maintains their shape. Then we have the exoskeleton. So our classic example of an exoskeleton are crustaceans. Right? We got a crab here. They have a hard shell on the outside of them. It provides where, that's where all their muscles are going to attach, all that sort of thing, provides leverage, provides protection. The downside of it is that once they grow, in order to get any larger, they need to shed that exoskeleton. Right? And that can be difficult, and it can leave them vulnerable for a while until they reharden. And then there's an endoskeleton. So an internal skeleton. You see this in echinoderms, so our sand dollars and our sea urchins. So now this is just the test or inner shell that is embedded in the body wall of the animal. What you would see in life is that this would be covered in spines and another layer of tissue. Same thing here. But it provides some protection for the internal organs and some support for the animal. So, a nervous system, right? The simplest one is a nerve, are these nerve rings or nerve nets, right? So, we've got a nerve net. It's just an entire net of nerve cells. Something happens on one end, it gets transmitted all the way through the organism, and a reaction happens, right? A little bit slightly more sophisticated is a nerve ring or a neural ring. So echinoderms like our sea star have that, where their signals will come in from this radial nerve, and then the neural ring will coordinate what sort of actions that the body takes. There are also central nervous systems. So we've got basically an insect sliced down the middle here. You've got a brain that is coordinating the overall actions of the organism. All right, so in this case, an insect. There are usually also some ganglion in each segment that will do some of the more automatic operations of each of those segments, but the overall actions are controlled by the brain. So, digestive tracts. These can also subdivide animals. So the simplest is the gastrovascular cavity again. So basically, you've got a mouth that also serves as the anus. Food goes in there, gets digested, what's left over is ejected back out. Same thing with our planaria here. It's got a muscular pharynx that it uses to eat. Food goes in through there, is digested in the gastrovascular cavity, and then is ejected out the same way. More sophisticated that is an alimentary canal, or a complete digestive tract. You can think of it as a tube within a tube. It might be more or less complex. We've got a very simple one in our annelid worm here. A little bit more complicated in our insect, and much more complicated in our bird. But in essence, it's just a tube that runs down through the middle of the organism and is responsible for digesting the food and then ejecting it out the anus. So to wrap up, animals are eukaryotic heterotrophs with no cell walls. Animals can be characterized by their body plan, their organization of tissues, germ layers, their symmetry, the type of embryonic development that they undergo, the type of body cavity, and their segmentation. So in the next tutorial, we're going to talk about the actual individual phyla of invertebrates, and we'll talk more about how each of these characteristics divide them up and show us how they're related. Thank you for listening. We'll see you in lab.